Welcome to your Microbiology It retention lesson. In this lesson, we are going to cover B cells and antibodies. First up is micro mind mapping. Get your pens and paper ready for that. Then we're going to review with some audio and then fill in workbook. Now, if you do not have a workbook, don't fret. Click the link in the description box to download your workbook so that you can participate. And then at the end, we are going to have practice problems to test your knowledge. More problems in the workbook. So go ahead, click the link in the description box, download the workbook, and let's microbiology it. It's time to micro mind map adaptive immunity, antibody production, and immune memory. Now, I'm not expecting you to write everything down that I have on the screen, but feel free to write down the major points from each major topic that I have. Then you can go back and listen to this again and write down any other things that stand out to you. So let's start with how B cells recognize and respond to antigens. So B cells carry surface antibodies called B cell receptors. That's BCR. And you see that in the micro mind map. Now, this allows them to detect specific antigens in the body. So, our micro mind map, main topic, B cells and antibody production. Number one, branch. Number one is B cells. Branch number two, antigen encounter. Number three, helper T cell activation. Number four, differentiation. So that's the start of our micro mind map. So once a B cell binds an antigen, the real work begins. After binding an antigen, the B cell internalizes it through endocytosis. Going to our micro mind map, number two, antigen encounter, we see branched from that is endocytosis. So that's very important. It breaks it down and then displays the antigenic peptide on the MHC class 2 molecule. And you see that in our micro mind map. So the B cell, which is a lymphocyte, has antibodies, are the receptors. The receptors bind to the antigen and engulf the antigen, and this is endocytosis. The antigen is digested and then digested into peptides. The peptides are then presented on the surface on a MHC2. Now, this presentation recruits the next player in the adaptive immune response, which is our branch number three, helper T cell. Helper T cell. So a T helper 2 cell recognizes the antigen on the MHC2 and releases cytokines that drive B cell proliferation. So you want to write down that cytokines are released and it stimulates B cell proliferation. Those are important. So now that the B cell can differentiate into powerful defenders, which would be the plasma membrane and memory B cells. So those are two things that you would want to write on our last branch, differentiation: plasma cells and B cells. Now, activated B cells divide into plasma cells, which secrete antibodies, and memory B cells, which respond rapidly if the antigen returns. Now, let's look at what antibodies actually do. Our next major topic is antibody function. Stemming from this, we have four branches: neutralization, branch number one; branch number two, opsonization; number three, circulation and coverage; and number four, immune efficiency. So antibodies neutralize toxins. They block pathogens from attaching to host cells and opsonize invaders, making them easier to engulf. So right there, we have our two branches covered: neutralization, opsonization. There is a blockage of the antigen and host interactions, and then the Toxins that may be present are surrounded by the antibodies, and this prevents the toxins from being attached to the host cell. Then we have opsonization, where the antibodies completely surround the pathogen, which helps in phagocytosis. Then we have circulation, which is our third branch, circulation and coverage, where the antibodies are traveling through the blood and lymph. And then we have immune efficiency, where there's target tagging and then facilitates rapid clearance, clearing all of the pathogens, antigens, and toxins out. The next, we will break down the major antibody classes. We have 5 of them. On our mind map, we have IgG, IgM, IgA, IgE, and IgD. Now, IgG, going back to that one, is the most abundant class in the serum. It has a Y-shaped structure and 2 identical binding sites that make it highly effective for neutralization. So, stemming from IgG, we have it's abundant. It has a Y shape with 2 identical binding sites. That's important. Now, there is another key player, and that is IgM. Now, IgM is the first responder. That's important to write down on our micro mind map. IgM forms a pentamer with 10 binding sites, giving it strong agglutination power, and makes it a potent complement activator. So on our mind map, we have it's a first responder, and it's a pentamer. It has 5 large molecules and a J chain. What's another one? I think those are the 2 major ones that you want to write down for now. So at the mucosal surface, a different antibody takes the lead, and that is IgA. Secretory IgA defends the mucosal surface like the gut, the lungs, saliva, and helps protect newborns through breast milk. So what you want to write here is that it's a dimer, and protects the mucosal surface. That's important. And newborn GI tract. And then also remember that it's found in saliva, tears, milk, and mucosal secretion. All right, IgE and IgD also play specialized roles, and we're going to cover these. Now, IgE binds to mast cells and eosinophils. It triggers histamine release and allergy and parasitic defenses. Then we have IgD, which is on the surface of B cells and serves as receptor and has low levels. There are low levels in the serum. So those are your immunoglobulin that you can write on your micro mind map. So now let's connect antibody production to where antigens enter the body. So we have our micro mind map. Next topic is antigen exposure and immune memory. Stemming from that, we have the spleen, lymphatic system, and mucosal exposure. The location of the antigen entry determines where the immune response begins. Blood antigens go to the spleen, the tissue antigens to the lymphatic, and the mucosal antigens to molds. So with spleen, we have 2 things stemming from it, 2 subtopics stemming from spleen. We have antigen enters the spleen, and then we have the antibodies that are produced, which are IgM, IgG, and serum IgA. With the lymphatic system, we have the antigen is carried to the lymph nodes, and that the same antibodies are produced, IgM, IgG, and serum IgA. Lastly, we have mucosal exposure, oral to intestinal moult, and in that there's a production of secretory IgA. Now, finally, let's compare the primary and secondary antibody responses. We have our micro mind map. Our last topic is primary versus secretary primary versus secondary response. Stemming from that is primary and then secondary. So the primary response relies on IgM and develops slowly. The secondary response, powered by memory cells, is faster, stronger, and often switches to IgG. So, what we're going to write on our micro mind map with primary is that plasma cells secrete IgM first, and it's slow in the initial response. And then with secondary, we have the memory B cells that's activated. It's faster and stronger faster and stronger in the response, and it has no help from the T helper cells. So those are the major things that you want to write down. Now that you've completed your micro mind map, use this time to review it. Adaptive immunity relies heavily on the ability of B lymphocytes to recognize antigens and produce structurally diverse antibodies. Each B cell carries surface receptors that are essentially membrane bound antibodies, allowing the cell to bind specific antigens with precision. Once a B cell encounters an antigen, it internalizes it through endocytosis, processes it into peptides, and presents those fragments on MHC class II molecules. This presentation recruits a T helper II cell whose cytokines stimulate the B cell to proliferate and differentiate into plasma cells. These plasma cells can generate thousands of antibody molecules, while a portion of activated B cells develop into memory cells that persist long term, enabling a faster and more effective response upon re-exposure to the same pathogen. Antibody structure underlies their functional diversity. The most abundant class in serum, IgG, contains 4 polypeptide chains arranged in a characteristic Y-shaped structure stabilized by disulfide bonds. Its 2 identical antigen binding sites allow it to bind the same epitope twice, enhancing neutralization and opsonization. Other immunoglobulin classes differ primarily in the amino acid sequence of their heavy chain constant domains, which defines their role in distinct immune environments. IgM, the first antibody produced during a primary response, is notable for its pentrameric structure containing up to 10 antigen binding sites, giving it strong agglutination capabilities and efficient complement activation. In contrast, IgA predominates at mucosal surfaces, often in a dimeric form equipped with a secretory component that protects it from degradation and plays a crucial role in immunity within the gastrointestinal, respiratory, and genitourinary tracts. Some immunoglobulin classes are specialized for particular challenges. IgE, although present in very low concentrations in serum, binds strongly to mast cells and eosinophils through its Fc region. When antigens cross-link the IgE molecules bound to these cells, it triggers degranulation and the release of histamine, serotonin, and other inflammatory mediators, an essential defense against parasitic infections, but also the basis of allergic reactions. IgD, found mostly on naive and memory B cells, functions as a receptor during early stages of B cell activation, even though it has no uniquely defined effector role in serum. The location of antigen exposure also influences the type of immune response generated. Blood-borne antigens are filtered through the spleen, inducing production of IgM, IgG, and serum IgA. Now, antigens introduced through the skin or peripheral tissues travel through lymphatics to regional lymph nodes, where a similar antibody pattern is produced. In contrast, antigens encountered at mucosal surfaces stimulate mucosa associated lymphoid tissue, or MALT, leads primarily to the secretion of IgA. Across all these scenarios, a primary antibody response is characterized by initial IgM secretion from short lived plasma cells, whereas a secondary response relies on memory B cells that rapidly expand and undergo class switching, most commonly to IgG, producing a faster, stronger, and more specialized defense with no need for renewed T cell help.